Hi, I'm Sean, and I'm going to talk about G protein coupled receptor signaling with heterotrimeric G proteins. And I'm going to talk about three main classes of G protein signaling, even though there are 4 or 5, I guess. So, I have a cartoon of a cell drawn here. This is the cell membrane. And then the black that you see, these are the G protein coupled receptors, okay? So I'm going to talk about GQ, GS, and GI, G protein coupled receptor signaling. These two here, GS and GI, represent G stimulatory and G inhibitory signaling. So I did green and red as in stop and go. So these are with respect to adenylate cyclase, which I have drawn here in pink, pinkish purple. And then GQ, just think about this one as calcium, like a different excitatory pathway towards calcium, especially for like smooth muscle contraction, okay? If this isn't smooth muscles. So you're probably wondering what the letter G means. G stands for guanosine. And guanosine is a nucleotide. It's a purine that is used in our DNA. So I'm going to quick draw this real quick. So for guanosine, you have a ribose. Sorry. You have a ribose backbone. This goes up to the actual nucleotide. This goes up over to where the phosphate groups are. If it's in DNA, there's just one phosphate and then another nucleoside, I think is what the correct term is. If this is ATP, there's 3 phosphates here. If it's ADP, there's 2. And if it's AMP, there's just 1, if it's not cyclic, and I'll get to that later. I'm going to quick draw the actual nucleotide. Generally, nitrogen is blue in chemistry. It's just kind of what they picked. I think it's because the sky is 70% nitrogen, our hemisphere is 70% nitrogen. So you have a nitrogen here, you have a nitrogen here, and then you'll have, let's see, black carbon is, I think, black. So these go over to a carbon. Jeez, my black marker is running out. So this is a carbon here, and then actually a carbon and 2 hydrogens, which we don't draw. When we draw just a backbone structure like this. And these go over to a carbon each, and these 2 carbons are connected to each other by a double bond, and then they're also connected over to, on the bottom case here, it's not a double bond actually here. So that's connected to another nitrogen. Then that nitrogen is connected over to a carbon. So it's like nitrogens are actually every other kind of here. So this is a carbon, then it goes up over to a nitrogen. So the ring on the right has 6 atomic components. Forming a hexagon. And then we have a bond up to an amino group here. If this is actually, no, I'm sorry, that's incorrect. We actually have an oxygen up on top. So it's a double bond, actually. I'm trying to draw another line here. And oxygen would be red in the chemical format here. So that's oxygen. Then we have at 4 o'clock, if you want to think about this like a clock, we have an amino group down here that connects to a nitrogen. A nitrogen with 2 hydrogens on it, satisfying the octet for nitrogen, if you know anything about chemistry, which still keeps it neutrally charged. The bone electron pair still hangs out by itself. Okay? Actually, these have hydrogens on them as well. These nitrogens do, but we don't usually draw them in chemistry. And then phosphates generally yellow, I believe, if I remember correctly. So in this case, though, I'm just going to draw circles just to save time here, since I only get 29 minutes on this. And this has a charge of, I'll use red because it's negative, a charge of -4. This is an oxygen here, by the way. Okay? So this is GTP, okay? GDP would have just 2 phosphates instead of 3 here. So GTP is guanine triphosphate, or guanosine triphosphate, okay? So if I took a phosphate off, if I cut one off, if I hydrolyzed it, used water to break these apart and then create an OH group instead, then what I would be doing is making it negatively 3 charged. It's a little more stable than GTP. Usually if a molecule is negatively charged like this, it's not going to be really stable, but these phosphate groups actually do have oxygens connected between these phosphates, and an oxygen here and an oxygen there. So an oxygen can have a double bond. If it's going to have a single bond, it's got to be negatively charged, or it has to have a proton connected to it. But that gives it a little bit of entropic flexibility, which helps, and that's called resonance, which keeps this together. But still, it's a very high energy molecule because it's negatively 4 charged. It's a, what is that, a quadrivalent anion. So this is GTP, and so the G comes from guanosine, which is, let's see, what color should I use? Let's pick any color, I guess. G represents this area right here. That's guanosine. Now adenine, which I'm not going to draw, but adenine is a little bit different. It's also purine. And now in the case of ATP, this would be with adenine instead of guanine. And in that case, you have an amino group, which is this kind of group here at 12 o'clock, where the oxygen is, and you don't have any other differences in it. So this part, it doesn't exist here, and this part is just an amino group in the case of adenine triphosphate. It looks pretty much identical to that. Oh, I didn't actually draw, my bad. I didn't draw the other parts. This is actually a ribose backbone, which means it has an OH connected to each carbon, pretty much. OH, OH. So this part's polar in the molecule. This part, I guess, might be polar, I guess. Yeah, I would think so, maybe. And then this part here, it would normally be polar, but it's connected to phosphates, so it's ionic. So it interacts pretty well with water, but it's also aromatic. And I didn't draw the double bonds here, speaking of aromatic. We have a double bond that kind of alternates between these 2 nitrogens because, well, if you think about it, nitrogen usually wants to have 3, but this carbon also wants to have 4 connections to it. It's got a hydrogen, but it's also got to basically alternate the double bond back and forth to share between these. And this has got a double bond here, so it's kind of like it actually alternates in this structure. In this case, let's see, this nitrogen needs 1, and so we could draw a double bond here, which kind of probably jumps around in time. If it's an adenine instead of a guanine, there's actually 3 double bonds in this part if you include this double bond here. So I basically just told you about the structure of guanosine triphosphate, and that is a molecule that binds to these proteins. Actually, I have them illustrated brown here. Guanosine triphosphate can bind, or guanosine diphosphate. which is pretty much the same molecule. The only difference is the number of phosphates, okay? The triphosphate's a higher energy molecule, just like ATP is the energy currency of the cell. When the triphosphate's bound to G protein, it's active. When the GDP, the diphosphate version is bound, then it is not active. But it's either GTP or GDP usually that's bound to that protein, and then that determines whether it's active or not. And that's how GTPases work. So GTPase means that it takes GTP and converts it to GDP. That's what that means. It cuts off that phosphate group, or it turns it into I guess GMP, but in this case it's just GDP we're talking about. It cuts off that one phosphate group using a water molecule by hydrolysis is what it's called. And that's an energetically favorable pathway. You don't need to add energy because the energy is coming from the intrinsic energy in this bond from the charges, but resonance is helping offset it a little bit, okay? So when you do that, you change the state and shape of a protein. So once it hydrolyzes that GTP, which is usually aided by, in this case, the receptor, which acts like a guanine nucleotide exchange, which can actually be catalyzed by proteins called regulators of G protein coupled signaling, RGS, and there's like I think 25 in the mammalian genome. But if those bind to it, it actually causes it to accelerate, I think by like a factor of 30, according to my cell biology textbook, 30 times as fast, causes to hydrolyze that to convert to GDP. But when it does, that GTPase is no longer active. But while it's active, it does some kind of function. It could be a number of functions in the cell. There's different GTPases everywhere. There's one that's even involved in like nuclear transport, which is called, I think, RAN, GTPase. But then there's also GTPase activating proteins that act upon those monomeric GTPases. In this case, this is trimeric, but still these RGSs that I just talked about, they're basically the equivalent of a gap, which is a GTPase activating protein that causes it to come to an end, activity-wise, because it causes it to hydrolyze that GTP to GDP, okay? A guanine nucleotide exchange factor for GTPases is a molecule that exchanges, it takes a GTP and gives it to the G protein instead of that GDP molecule. So it gives it back that higher energy and then allows the molecule to change conformation and then go do some function until some RGS or some gap comes along and then tells it to quick convert it to GDP again. Once it does that, that inactivates that G protein, whatever its function was, it stops. Even though functionally it's a GTPase. But so basically it has two functions, one that limits itself and then one that does something in the cell. So that's how your cell thinks if you want to anthropomorphize the cell, okay? So I'm going to start talking here about the G protein coupled receptor here, that's the GS pathway. And so if I was going to talk about this in terms of the autonomic nervous system, which I just talked about earlier, and I was just going to talk about norepinephrine or epinephrine and acetylcholine. So GS is your beta receptor. So this could be a beta 1, 2, or 3 receptor. This could be an alpha 2 receptor here, or it could also be a muscarinic 2 or 4. If it's an even number of muscarinic, then it's going to be inhibitory signaling. Beta can actually also act upon, beta 2 I should say, can also act upon the G inhibitory pathway, some people think, okay? Then there's the GQ pathway that activates calcium and smooth muscle contraction. And this can be mediated by your odd-numbered muscarinic receptors, muscarinic receptor 1, 3, and 5. And 5 is usually found in the central nervous system, so usually it's 2 and 4 if you're not crossing the blood-brain barrier with drugs or anything, you're just staying in your body. So in that case, so if it's not like like you have different like they're called parasympathomimetics. If it has so you have, oops, let's use blue actually. So acetylcholine, I'm not going to draw up the whole structure of acetylcholine, but acetylcholine's got a nitrogen in it, and if you have a structure in which you have 4 different bonds on that nitrogen, for example, that nitrogen is going to be positively charged. And I guess I don't know what color to use actually. We'll just use blue, I guess. We'll use a plus, okay? So if that's positively charged, it's ionic, and it's not as likely to cross the blood-brain barrier. So drugs that have nitrogen with 4 connections to it are going to be positively charged, just like ammonium is positively charged. It's not likely to move across the blood-brain barrier, but if it's got a lot of hydrophobic groups and it's only got 3, it might move across the blood-brain barrier. So I'm just mostly confining what I'm talking about to molecules that don't cross the blood-brain barrier, like parasympathomimetics that just operate in your peripheral nervous system. Those drugs, if they bind to a muscarinic 1, 2, or 1, 3, or 5, probably just 1 or 3, because 5 is in your central nervous system. If it binds to a 1 or a 3, and it causes activation of the GQ receptor here, which would be muscarinic 1, 3, or maybe 5, maybe probably 1 or 3, or in the case of your adrenergic nervous system, it would be activating the alpha-1 receptor, because that's also using GQ signaling. Then it would be activating this pathway. In the case of drugs like isoproterenol, metoprolol, levobitorol, different drugs that activate your beta-2 receptor, it's going to be activating the stimulatory pathway, or even ones that just activate the beta-1 receptor like terbutaline, and well, I guess isoproterenol activates beta-1 and 2. If it inhibits, it could be like a beta blocker like propranolol, or just some of the other alols that aren't specific for just beta-1. Or even if it's specific like nebivolol, it's still activating still inhibiting the GS pathway. Inhibitory 2 and 4, like molecules that inhibit muscarinic 2 or 4 receptors, or are inhibiting your alpha-2 receptor. I'm trying to think of one. I guess phenoxybenzamine irreversibly changes this receptor, or let's see, phentolamine also does too. But I don't know that I'd want to take at least the first drug because it's an alkylating agent, and it's only used for pheochromocytoma. I think, actually I'm not 100% sure about that now. Never mind, forget about that. So now I'm just going to talk about each signaling pathway. So starting with the stimulatory pathway, when this gets agonized, so this is a beta-2 receptor, you have agonist bind, and in this case it's mostly epinephrine, because norepinephrine mostly just binds to the beta-1 receptor. It doesn't really bind as much to the beta-2 receptor. So let's say epinephrine binds to beta-2 here. Beta-2 immediately acts as a guanine nucleotide exchange factor towards the G protein here, GS alpha. You have a beta and a gamma subunit attached to it as well. When it gets activated, those dissociate. And when they dissociate, that G alpha stimulatory protein goes over, binds to adenylate cyclase. Adenylate cyclase accepts an ATP molecule. Remember I was talking about this, except in this case it's got an amino group up here instead of the oxygen, and there's nothing here. So you have 3 phosphates connected to this. It's very high energy. It uses that energy to basically cause a cyclic bond, and this is called adenine 3,5 monophosphate. So it actually only has 1 phosphate group that's negatively 2 charged. I think negatively 2? Maybe negatively 1. I'm trying to think. I think negatively 2. No, negatively 1, because it's connecting it back to the sugar backbone. So it takes this and it connects it back over to the 3 position here, to this. It it gets rid of this OH group, gets rid of the hydrogen at least, and it actually attaches it to the phosphate group instead. So this phosphate connects to this, and it causes cyclic. That's why they call it cyclic adenine monophosphate. But even though it's 3 and 5 positions are connected, when it does that, this is a distinct molecule from AMP, ADP, or ATP, and it has different effects in the cell. It can bind specifically to another protein that's called protein kinase A, also known as cyclic AMP-dependent protein kinase. Once 2 molecules bind to each subunit, and I believe it exists in a quatrimer. These are, I think, ACAPs, which are regulatory protein kinase A molecules that bind to them as well. Once 2 of these cyclic AMPs, I'll just draw that out. Cyclic AMPs bind to each of these, at least 2, maybe it's more than that. When they bind to that, that causes activation of this molecule by releasing these red subunits here that are inhibiting it. When those are released, each of those subunits can go and become a kinase. And a kinase takes ATP, and let's just write ATP here. ATP. And then this is also cyclic AMP, actually. Cyclic AMP. That converts ATP, though, over to AMP, basically. But it takes one of the phosphate groups and it adds it, or ATP or ADP, I'm not sure actually what the reactant is. But it takes it and it adds a phosphate group, which I've drawn in purple here. Even though if I were to keep my format, I guess I would use, I think, yellow. But I'm not going to worry about it, I guess. So this protein here is called CREB, CREB, which is cyclic AMP response element binding protein. So this protein goes and it binds to certain DNA sequences that whatever it binds to here, this macromolecule, it binds to certain DNA sequences in your DNA, and when it does, that causes other machinery to come and bind to it, and then that activates transcription of certain genes. Usually there's numerous genes in series with each other that get transcribed. I think it might be like one of those enhancer elements, but it binds and it activates genes downstream of it. Those genes get transcribed. This takes several hours. It takes like maybe 2, 3, or more hours for these proteins to be transcribed, but eventually they get translated into protein, or maybe if it's a microRNA, it gets processed and then it starts doing different things in the cell. But those are delayed responses. Protein kinase A also does other things. Oh, I didn't mention, this is CBP, which is CREB binding protein. It's just basically another protein that binds to it that's a co-activator with it. But also, protein kinase A here, which I will actually write out. Protein kinase A goes, and it also does other functions depending on which cell you're in. If you're in the liver, it's going to want to cause like glycogenolysis and gluconeogenesis. It wants to create glucose. It wants to break down glycogen into glucose, and it wants to release it into the cell for an emergency to make sure that you have what you need if your sympathetic nervous system is being activated. Remember, sympathetic nervous system activates the beta 1, 2, and 3 receptors, and also the alpha 1 and 2 receptors. And when that happens, those execute different programs in whatever cells these receptors are located in. Okay, in the liver, you're going to want to basically want to release glucose, and you're also going to want to create glucose and break down glycogen in those moments. So that's what that does in those cells. In other cells like smooth muscle, protein kinase A inhibits the myosin light chain kinase, which inhibits smooth muscle contraction, which is why in your internal sphincter, in your just below your bladder and your urethra, if you're in a fight or flight response, you're not as likely to pee at that moment, just because it's causing contraction of that. So, wait. I think about what I just said. So yeah, if it's an alpha receptor, no, that's why I got confused for a second. So actually, never mind. Scratch what I just said. Because protein kinase A. So in other cells, like your vascular smooth muscle cells, for example, like in your muscle, you're going to want to have vascular dilation. So you're going to want to relax your smooth muscle in your muscle cells, because beta 2 doesn't have to be everywhere in your body. It's just in the places where your body wants it to be, determined by your DNA and your developmental pattern, your epigenetics, all that stuff, that really complicated program where you start from a zygote and end up being a fully formed human. It causes these receptors to just be expressed on the cells that it wants, and that's in your muscle cells. So like your skeletal muscle cells, if you're using them especially, probably, it's going to want to have blood going to those cells. So it's going to want to relax the smooth muscle so that blood goes to those places instead of the other places. Whereas your alpha-1 receptor, it's going to want to squeeze those places. Like, I don't need my fingers as much, I don't need my digestive tract as much, and some of the other places, so we don't need blood going there, we want to shunt it to our muscles, right? So the beta 2 receptor helps get blood over to your muscles and also tells your muscles to take up glucose, etc., using the protein kinase pathway and the cyclic AMP pathway. So when it does that, protein kinase A is going to want to phosphorylate myosin light chain kinase, inhibit it so it's not activating your myosin light chain by phosphorylating it, and in that case it causes smooth muscle relaxation. I have to start another video because I'm towards the end of this, but I'll do a second video and we'll talk about GI and GQ signaling, okay?